different structures together. It fills interstitial spaces. So adipose tissue can store energy. It can be used for energy storage. The blood cells are considered to be a connective tissue, and so they're able to transport materials throughout the body. Epithelia and connective tissue often work in very close and coordinated fashions. The other two types of tissue, which are each going to have their own lecture, are neural tissue. This is the tissue that carries electrical impulse that are solely restricted to the nervous system. And then there is muscle tissue. And muscle tissue, there are three types of muscle tissue. As I said yesterday, there's skeletal, cardiac, and smooth muscle tissue, each which has very distinct characteristics. These other two types of tissue are both excitable tissue. These are excitable cells, and these are non-excitable cells. So on to it. Here is an example of how you might break down material that I'm talking about. Okay, so and I did this for you so you can get a sense. Going through my lecture notes, I broke it down into an outline form, a schematic outline form. And this is the kind of thing that you may want to be able to do in your own notes. I did it for you here in this first or second lecture so that you can get a sense of what it would look like. Probably kindergarten stuff for most of you, but just so that you see what an appropriate framework is. This is everything we're going to cover today. Yep, everything we're going to cover. We're not going to go through this in much detail, but that is an outline of the tissue types. So as I had mentioned, their epithelial cells are cells covering various surfaces, and they're also they form glands. So in epithelia, the category of epithelia, we talked about yesterday the fact that there are squamous, or the squished cells, the cuboidal, and then the columnar. That's three categories. And then within each of those, there are three subcategories, which we're going to go through one at a time. And then in glands, there are endocrine glands, which will have its own whole lecture. And then there are exocrine glands, and we're going to talk about the distinction there and the different types of exocrine glands. In connective tissue, there are three broad categories. There's connective tissue proper, and all of these names are very descriptive. So it is what the name implies. It's connective tissue that's actually connecting stuff, that's holding some sort of, giving some sort of structural support, a structural support element to the organ that it finds itself in. These break down into dense and loose connective tissues with three subcategories that we're going to highlight today. And across this, there are 9 cell types that can form any of these dense or loose connective tissues. And these are melanocytes, mast cells, lymphocytes, macrophages, fibroblasts, fibrocytes, macrophages, adipocytes, and mesenchymal cells. We'll talk about some, not all of those. Then following connective tissue proper is these fluid connective tissues. And these are the plasma, so the fluid in your blood, the fluid component of your blood, the interstitial fluid, that is the broth that surrounds each of our cells, and then the lymph in the lymphatic system. And then there are the supportive connective tissues. These are the connective tissues that essentially form your skeletal system and other protective elements like your trachea and the pinnas of your ears, so cartilage and bone. Right? And then finally, there'll be the muscle and neural tissue, and then we'll finally get to defining a membrane, what a membrane actually is. And this is not a plasma membrane. This is not the single lipid bilayer of an individual cell. This is a tissue membrane, okay? So a higher order up, a membrane composed of multiple cells. Just to give you the punchline now, a membrane is a layer of epithelium on top of a layer of connective tissue. That's what a membrane is. A layer of epithelium on top of a layer of connective tissue. We'll talk about the 4 membrane types. All right. So all of these cell types embryologically derive from one of the 3 primary germ layers. So here is a little embryo implanted on the placenta, forming a placenta on the inside of the uterus. These 3 layers are endoderm. The endoderm ends up being sort of the interior of the coelomic cavity, the lining of the mucous membrane lining of the digestive and respiratory tracts. And then the mesoderm is the mesenchymal cells, the muscle cells, the heart, the kidney, blood, the bone. And then the ectoderm is where the epidermis and nervous system derive from. Okay? So that is the embryologic origin of these. Let's look at the generalized structure of an epithelial cell. So the tissue categories that I showed in the first slide there, the outline, were within epithelia. There are epithelial cells, which are the covering cells of a layer, or some sort of layer, and then the glands. And glands produce secretions. What is a characteristic that all epithelial, what are the characteristics that all epithelial cells have? Well, there's a cellularity. By cellularity, we mean there are junctions between them. There are cells. It's like in an apartment building, you have different rooms, or a classroom, there's rooms, and you can think of them as cells. There are walls dividing each of the compartments. That's the notion of cellularity. Seems pretty obvious, but it is certainly a characteristic of epithelium. For example, certain connective tissues are not going to have the same sort of cellularity, like fluid connective tissues. The red blood cells do not join together. Okay, they're individual cells. Cellularity doesn't mean that they are cells. They are cells. Cellularity means that those cells adjoin one another in forming a superstructure as an ensemble. Epithelia has polarity. Is this new to anybody? Has anybody here had this material before? Some of you are familiar with this concept. So there's the notion that a cell has an apical and a basolateral surface, a top and a bottom. There's sort of a directionality to epithelial cells. Not all cells are like that. And there's a functional, so going back to Louis Sullivan's original form ever follows function, there is a rationale for this, a functional rationale for this structure in these epithelial cells, and we'll get to that in a moment. In fact, it derives from the fact that epithelial cells have cellular products. They make stuff. They have secretions. We'll see how that works in a moment. They have attachment. Epithelial cells have attachment to a basement membrane. So here is an epithelial cell. Here's some sort of connective tissue that's adjacent to the basolateral surface. This this underlying connective tissue forms a basement membrane. Okay, there's a basement membrane. that plasma membrane in that cell adheres to this underlying connective tissue, right? So that basolateral surface is composed of a basement membrane. It attaches to something. Epithelial cells attach to something. They're not just floating around. They are avascular. This relates to the cellularity. It's avascular, meaning blood vessels don't penetrate up into epithelial tissue. Even the stratified epithelium that has multiple layers, you're not going to have blood vessels working your way up into a stratified epithelium. And that's because of these junctions here that we're going to go through in a bit that form the cellularity, that adhere these cells to one another. So they're avascular, and that has important consequences for the function of epithelium. And they're regenerative. You can damage them, or they can just die, senesce, and they can be replaced and regenerated. Not all tissue types are like that. Neural cells have a very hard time regenerating, for example. What does it do? Well, it protects things, right? It's a coating, so it serves to protect. Because of this cellularity, because of the ability of the lateral surfaces of these epithelium to adhere to one another, it's able to control the permeability of that layer of epithelium. So over here, beyond the apical surface, there's the outside world. There's the lumen of your intestines, or of a duct, a tube inside of the kidneys, or the surface of your skin, whatever. There's the outside world, and that epithelium is sort of the gatekeeper saying what can pass through this layer of cells and what cannot, right? So it controls the permeability across this junction, right? Oftentimes, epithelial cells are invested with neural tissue and can provide sensation, and they make specialized secretion. Glandular epithelia make all kinds of secretions, from earwax to sweat to hair oil to et cetera, and on and on. Okay? I think that's all I need to go through. The surface, the apical surface, is often coated in these little finger-like projections, which increase the surface area of that. Why? Just given what I've told you already, not expecting you to know this, but see if you can stretch your minds. Why might the apical surface have all these finger-like projections?
It helps with absorption. There's more like surface area.
There's more surface area. It helps with absorption and secretion, right? So that is a membrane, that is a plasma membrane that is involved in an exchange with the outside world. And so the more surface area you can pack into a smaller volume, the more efficient this cell is going to be at doing the job it needs to do. Okay? When we get to lungs, I'm going to talk more about surface area and volume. It's pretty interesting when you really begin to think of it. Okay, these connections that give rise to the cellularity. There's a number of types of connections that we're going to have here. And there's 4 categories that I have here. There are these tight junctions. So right at the plasma membrane, right up at the top, where at the apical surface, where these villi or cilia or whatever you have, depending upon the cell type, it's almost like a Ziploc type of connection. All right, a totally tight seal, an adhesion belt, happens between the 2 adjacent cells that excludes the passage of anything through this, this junction right here. Okay, so we have a tight junction. We're not going to go into the underlying protein structure of that, but it's pretty interesting how it works and how it connects with the underlying cytoskeletal elements and how that affects signaling in the cell, and etc. There's a lot to know there. It's pretty interesting. Next are these gap junctions. In fact, before we get to gap junctions, because I think they're the coolest, we'll go to these desmosomes and hemidesmosomes. Did anybody take shop class in high school? Anyone ever had shop class? Did you ever get to use a spot welder? No? Well, a spot welder is a tool that you use to take two pieces of sheet metal, and there's these two electrodes that go, you stick it in between, it goes bzz, and it just sticks the two sheets of metal together right at the little spot where the welding happens. That's exactly like desmosomes. They're just little spot welds. They're little points where the desmosomes are points where adjacent cells are just adhered to one another. So these tight junctions are giving the impermeability of the membrane, of the layer of epithelium, and these desmosomes are stabilizing that structure beneath that tight junction. Okay? So if they weren't there, these cells would be zipped here, but adjacent cells could be moving with respect to one another and could shear open those tight junctions. So these tight the desmosomes that give these little focal adhesion points stabilize the structure of the adjacent cells. And then there are these hemidesmosomes. A hemidesmosome, what does the word hemi mean? It doesn't mean just big truck. What does it mean? A hemi means half, just like a hemisphere, half of the sphere. So it's a desmosome, but hemi because it's only on one side. You know, it's just in this cell. It's not actually adhering to another epithelial cell. Beneath, it's adhering to the basement membrane of connective tissue. Okay? So it's a hemidesmosome on the bottom, on the basolateral surface, and then desmosomes on the side. Finally, gap junctions. Gap junctions. Has anybody here ever sailed before? There must be some sailors. No? Rivets, like in a sail. Like you have a big piece of canvas, and you put a little a rivet, a grommet. Is that the right word? Maybe a grommet's the word I'm thinking of. The big metal ring. There's a hole through it. You put the ring, the rope, and tie the sheet down to the boom. That's what a gap junction is. It's basically a little grommet that adheres to adjacent cells. But unlike a desmosome, whose primary purpose is strength and also communication, so there are signaling events that are happening here because these desmosomes are connected by cytoskeletal elements, which is a much more complex story. Gap junction, their job is to create a syncytium, meaning that the cytoplasm of adjacent cells can exchange elements that pass through the gap junctions. So adjacent cells can work in a coordinated fashion. Whatever's happening in the cytosol of one can be selectively shared with the cytosol of an adjacent cell. Okay? The story of gap junctions, in my opinion, goes far beyond that, particularly with their role in neurons, but we're going to leave it there for now. But that, for people who are really interested, you could devote your entire careers to what's on this page. Okay. We saw this yesterday, just to review. Classifying into simple and stratified. Simple is a single layer, a monolayer. Here there's stacked layers, squamous, these are squamous, squashed down, flattened cells, cuboidal, cube-shaped, and columnar. And then there are the two other types. We have a pseudo-stratified columnar. This looks stratified, but it's not. There's actually only single layers here, but the cells don't rise evenly. They're sort of like twisted around one another. So when you take a tissue section, it looks like this, but it's actually, it is a simple layer of cells. So they call it pseudo-stratified, ciliated. And then transitional epithelia is it's an epithelium unique to the lining of the bladder. And what might you guess would be a good characteristic of the lining of your bladder? If you were going to design a covering for the inside of your bladder, what would you build into it? What do you think? Just guess. Doesn't matter. Say whatever you want.
Not too rigid so that different structures can.
Yeah, not very rigid for sure. Why? What happens in the bladder?
Nitrogen staying out.
That's in the
Sorry.
kidney. Yeah, you're right, but you're on the right track.
Excretion.
Excretion. The bladder is primarily for storage of urine, right? We store urine in the bladder. And say you made the dubious choice to sign up for a 2-hour straight lecture class, and you drank an enormous amount of tea first thing in the morning. What's going to happen to you by hour 2? Your bladder gets full. Your bladder stretches, right? Your bladder stretches. And so we want transitional epithelium to be able to stretch and then come right back to the same shape. So it's really stretchable, much more so than any of the other tissue types. All right. Glands. So let's, first of all, define the distinction between secretion and excretion. Secretion is the production of some kind of cellular product that we want to keep. We have a use for. It's doing something for us. Excretion is the removal of a waste product that is not useful to us, that we are trying to eliminate through the urine or by other means into our colon. All right. So the two broad categories of glandular epithelia are endocrine and exocrine. Endocrine glands release hormones into the interstitial fluid. There are no ducts in endocrine glands. Right? These hormones are released into the interstitium and can either act in the tissue that they've been released into, or they can enter the bloodstream from that interstitial fluid. All right? Exocrine glands produce secretions into ducts onto the surface of the epithelium, which then forms a duct. All right? We have 3 types: merocrine, apocrine, and holocrine secretion. We're going to go through those. So here is a duct, an exocrine gland. This is some sort of exocrine gland with this arborized ductwork here. And these epithelial cells are exocytosing their cellular product onto the surface of the cell, which is in the lumen of a glandular duct. All right. So the distinction, the functional distinction, or maybe I guess the word is mechanistic distinction between merocrine, apocrine, and holocrine is in how radical a process these cells go through to disgorge their cellular products. So the first is the most gentle. It's merocrine secretion. These secretions are made in the Golgi apparatus in the cell, and they're released by little vesicles. So these bubbles, the Golgi apparatus here produces these contents, these cellular secretions, these products, surrounds them in the vesicle, in the membrane, in a membrane little bubble, which is a pinched off part of the Golgi apparatus. So they happen inside the lumen of the Golgi. Parts of the Golgi pinch off and are released as bubbles, little space pods, into the cell, and then they merge with the external plasma membrane. Those membrane vesicles merge, and that the inside of the vesicle becomes part of the outside of the plasma membrane. And what was inside that vesicle is then released out into the duct. All right? An example of this is sweat. Sweat. Just like full body, everywhere, sweat of your brow, sweat. All right? Designed to cool you off. Designed to release mostly just water, but maybe some salt, etc. That's merocrine secretion. Apocrine secretion is also produced in the Golgi. However, rather than exocytosing a little bubble and keeping the plasma membrane, right? Because in merocrine, we have the plasma membrane. Oops, I'm doing a different color. This chalk sucks. The vesicle comes up towards the plasma membrane, fuses, and then we have this. Right? The membrane doesn't go anywhere. All the stuff that was inside is outside, but we've retained that plasma membrane. Okay? And we can recycle the plasma membrane back down to the Golgi. In apocrine, what happens is, damn it all, we just break off the top and get rid of the whole top chunk, the whole apical surface of the cell. All right? So we lose plasma membrane in apocrine secretion. An example is mammary glands. Mammary glands. So we see here, here's a cell. It's beginning to pinch off. This chunk comes out and begins to break down, and it regrows and continues this cycle. Why? Why would this be a clever strategy for releasing milk in a mammary gland?
This is more efficient.
It's more efficient. Why? Okay.
Because those vesicles are really small, and if they want to get rid of the same amount of stuff inside the cell, it needs a lot of vesicles. But in this process, they need only one time, and it stays for all.
Okay. Excellent. So there's one reason. There's one reason. What else? Keep going, Tanner.
The lipid membrane is pretty potentially energetic.
Oh, nailed it. Thank you. Yes. So not only are we releasing fluid contents, water-soluble contents, we're releasing lipids as well, fat, which is what we want in milk. All right? So apocrine secretion has a more complex mixture of cellular products that are being released. Does that make sense? Finally, there's the nuclear option, holocrine secretion. This is by basically just the destruction of the cell. We release the entire cell, the whole cell blows up, releases its contents and are just replaced by stem cells. So apocrine can be, what's one difference in the two broad categories here? Not just the mechanistic, but what in the types of epithelial layers? What can you say? This is simple, and this is stratified. Okay. So this holocrine secretion needs to be stratified because the lower levels have stem cells that are constantly dividing. And the cells come up, blow to bits on the surface, and then are replaced from below. Sebaceous glands, sebum, skin oil, really waxy stuff, designed to help protect the surface of your skin. You know, lots of lipid component, all of this cell membrane, all of those lipids and the waxes inside that cell are released upon destruction of that cell. Okay. Also, the sebaceous glands, you can think of them as being on the surface of your body, right? This, even though this surface here is a tiny little tube that goes down into your skin, if you were a tiny little hiker, you could walk on the skin down into that tube. So that is the outside, like if you were a bacteria or something. So having a nice thick stratified layer there helps protect the skin. All right, so there are a bunch of structural classifications for the exocrine glands. So to help you remember those words, exocrine, exo, outside. It's releasing the contents into ducts that are kind of leading somehow out of the body, right? Ducts, into ducts that lead out onto the skin, into ducts that lead out into the lumen of your gut, which is kind of like the outside of your body, even though it's totally inside your body, right? You're a tube. And endocrine are releasing into the interstitium, which is genuinely in the body. Okay? That goes then into your blood. You cannot get there, get from an endocrine secretion to the outside world. So these glands break down into simple, and the next one is compound. And there's these 5 tissue types. I'm not going to test you on this, but I'm showing it to you. So tubular, coiled, branched, and then this alveolar or branched alveolar. So an alveolus is like a little pouch. It's like a cul-de-sac at the end of a tube. Right? These are different structures in the glands. And then these compound glands you have compound tubular, so a branch of tubes. You can have compound alveolar, so it's the tubes have alveolus on the end of them, and you can have tubulo-alveolar. It's all possible out there. Biology's taken advantage of all of it. On to connective tissues. Any questions on epithelium? I took way too much time on that. Nope. All right. 3 types: connective tissue proper, fluid, and supportive connective tissues. So they bind organs, they support, they protect. We've gone through all this. It's also immune protection. So your white blood cells are responsible, they're the primary agents of your immune system. The protection of invaders is taken care of by connective tissue. Movement, so bones act as the rigid levers that muscular tissue works on. Stores fat, calcium, and phosphorus. Fat in the adipose, calcium and phosphorus in the bone. Heat production, so brown fat, there's 2 types of fat, white and brown. Brown fat is enriched in mitochondria and it's found only in babies. This is how babies are able to enviously drop tons of chub so quickly because that fat that they have is not like the fat that you have after Christmas. It is, or whatever, it is brown fat that can be mobilized quite quickly. And because of the mitochondrial content of those cells, and produces heat. You ever seen, I have a young daughter, 2 kids, my daughter in particular, she does not like to wear clothes. And when I have like 3 sweaters on, I'm like this, she is just like a little radiator. Kids can often be like that. They'll run around outside. They don't seem to get bothered by the cold too badly. They have very high metabolism. They're producing a lot of heat from the brown fat. And transport in the blood. And then the basal lamina connects epithelium to the rest of the body. That basal lamina that we talked about with the epithelium is formed by connective tissue. All right, so here is a diagrammatic tree of the types of connective tissue, the proper, connective tissue proper, and then the specialized, the supportive and liquid connective tissues. And proper breaks up into loose and dense. There are 9 types. These are the cell types that I showed in that original outline. We're going to walk through them. Fibroblasts. This is by far the most common cell type in the body. Of the connective tissues. This is throughout all types of connective tissue proper. And these fibroblasts, they basically are producing mortar, cellular mortar, the mortar of the body. They're making various proteins and hyaluronic acid that act as cellular cement in extracellular matrix. They're moving around and creating different types of fibers. So fibrocytes, these are also extremely common because they're derived from fibroblasts, and that's also in all connective tissue proper. These guys lay down new material. These guys help maintain that material. So when you have, say, an injury, fibroblasts move in, lay down the hyaluronic acid and all the other types of proteinaceous materials, and then they mature into fibrocytes, which then maintain that tissue. Adipocytes, fat cells. Fat cells are essentially cells that are very little else besides a single, totally jam-packed vacuole of lipids, fat. All right, just big storage containers. Although they do have a role in the endocrine system. They produce a variety of hormones, leptin, ghrelin, that kind of stuff, which we may talk about, and I may have time to talk about later in the semester. But they've been a lot of research focused on the endocrine function of adipose tissue lately, particularly related to obesity and insulin resistance and metabolic disorder. Mesenchymal cells. So these are cells that get stimulated when there is some sort of injury or infection, something's wrong in a tissue. These are not terminally differentiated cells. Mesenchymal cells can differentiate themselves into a host of other, they're stem cells. They are stem cells, yeah. They can differentiate themselves into a host of other connective tissue cell types, such as fibroblasts, macrophages, etc. They're like the raw material cells of connective tissue. Macrophages, these things are amazing. Sometime when you're bored and you want to look at some incredible pictures on Google Images, just Google image macrophage electron photograph. There are these giant amoeba-like cells, the stuff of 1950s horror movies. It's big. They send out big pseudopodia. They search and destroy the invaders surrounding them, and eating them up. Damaged cells, pathogens, what have you. There's two types. There are some that stay in a tissue. They don't want to leave the specific tissue because they're reading various environmental cues in that tissue, and so they can migrate around in the tissue, but they're not leaving the tissue. And then there's free macrophages who are free to go anywhere in the body that they're needed. So, pretty cool thing. Yeah, here, let me just show you that. These are bacteria. These are some sort of bacteria there. And this macrophage is just clean in house. It's getting sort of attacked, but it doesn't care. It's just going to eat all that stuff. All right, mast cells. Mast cells are like they're like Paul Revere. They ring the alarm bell. And if there's a problem, they release chemical or a perceived problem. They release chemical signals that start an inflammation cascade that cause redness and swelling in a tissue, which dilation of the capillaries in that tissue, which brings more cells in, says there's something going on here. Histamine and heparin are the two primary chemical factors that are being released. They're not alone. Basophils are another type of leukocyte, actually a very rare type. There's not, they are in your body, but they're not the most common white blood cell that have overlapping functions to mast cells. Mast cells, so allergy is a dysfunction in the antigen-presenting cells in the body, but the downstream consequence is that mast cells are what are getting activated. And so when you take an antihistamine is not solving the problem, but it's solving a symptom for you. It's dealing with the histamine that's been released by these mast cells. The histamine is an inflammatory chemical. It's not removing the source of the irritation, it's just sort of knocking out the alarm bell. Okay? Make sense? Lymphocytes. These are white blood cells. There's a whole host of them. We're going to talk about them in lab and in the lecture down the road. These cells can develop into plasmacytes of different types, leukocytes, that are going to make antibodies and do all kinds of other things. There are ones that don't produce antibodies. Lymphocytes. Microphages. So, similar to macrophages, but they're small. They're phagocytic blood cells that work in coordination with macrophage and mast cells when there is some sort of problem. For example, eosinophils, neutrophils, and basophils. This is another type of white blood cell. These are all microphages is a broader category of white blood cells. Not all white blood cells are microphages, however. Lymphocyte is the other category of white blood cells. So we have lymphocytes that can become monocytes, T and B cells, etc. And then eosinophils, neutrophils, and basophils, which is on the other side. They are phagocytic, and they can produce types of antibodies. For example, eosinophils produce IgE, which is the antibody type of antibody that's responsible for allergy in the body. Melanocytes. These are cells that are making melanin. They're also pseudopodic cells. We're going to talk about them later in the lecture if I can get moving fast enough here. they make melanin, which is a chromophore that absorbs UV radiation. This gives your skin its color. They invest and work their way into the stratum germinativum of the basement layer of your epidermis. It is this cell type that, when it has enough damage from UV radiation from sun, becomes melanoma, which is the aggressive, the bad type of skin cancer. We'll talk about skin cancer at the end of the lecture, though, presuming I get there. All right. What kind of fibers are these fibroblasts making, or fibrocytes? Well, we have collagen. There are actually many types of collagen in the body. The last slide of the day in this lecture, I'm going to talk about a mutation in the collagen 7 gene. It's a straight, unbranched cable, much like a nautical hawser, one of those big ropes that you would tie up a tanker with. Very strong, not stretchy collagen. Reticular fibers. So here is some reticular fibers. These, unlike collagen, resist force in 3 dimensions. Imagine instead of a single rope that only restricts tension in 1 dimension, imagine like an octopus or one of those koosh balls, those things with all the little fibers that come out. Instead, it's not stretchy though. It restricts stretching in 3 dimensions. Okay, so this can make like the capsule, for example, to the kidney. The layer surrounding the outside of the kidney is made of reticular fibers. So it's thick, it's strong, it can resist tension in multiple directions, not just a single direction like a collagen fiber. And then elastic fibers are slightly branched. There are branches to elastic fibers, but they are not super common. And elastic fibers are real stretchy and kind of a little bit springy sometimes. All right, real stretchy, allows tissue, helps bring tissue back to its original shape after it's been deformed. All right, so this is a diagram of alveolar tissue. We talked about alveolar tissue yesterday. This is the tissue, connective tissue, equivalent of like a rubber sheet or something. Real stretchy, strong stuff. Maybe like lycra pants or something. I don't know. But this is showing a whole host of the different connective tissue cell types in a single connective tissue. So many connective tissue cell types in a single connective tissue. I hope I drove that home. We have where are the fibroblasts? So here's one. So all connective tissue proper has fibroblasts in it. So that's there. Fibroblasts are making some of this collagen. They're probably making some of this elastic fiber. You can have these are adipocytes, fat cells down in there. Here's some lymphocytes, white blood cell that maybe is a B cell or something. There's a bunch of immune cells throughout here, reticular fibers. In between this negative space, between everything in this slide, everything that's pink here, is called ground substance. Ground substance. It is clear, viscous, and colorless. Has anybody ever had one of those, like any kind of squishy thing, any kind of squishy toy that your older brother cut the head off of, and it like oozed this gooey stuff in it. That's the way I imagine ground substance to be. Honey is not quite the right description because honey will drip off of itself. This is like a goo that sort of sticks together. It's viscous, it's clear. It makes it really hard for bacteria to just cruise through the tissue. They have a hard time swimming through. It's not like water or like some sort of solution. It's like a thick, viscous stuff that's full of glycosaminoglycans, proteoglycans, and adhesive glycoproteins. I don't know what the next slide is. I actually imagine I'm going to talk about what those three are in the next slide. Maybe I will. Let's see. Do I? Yes. Great. As I said, I'm a carbohydrate biophysicist, and this is the kind of thing that I really get excited about. So we'll talk about it for a moment. GAGs. Glyco, the prefix glyco means sugar. It means a carbohydrate. It's a generic term for a sugar, okay, glyco. And amino is referring to proteins, amino acids, right? And then glycans. Glycan is just a word that means some kind of sugar chain. In general, a sugar, and I'm not talking about table sugar, there's a whole wide world, a big tree of different structural analogs that are all carbohydrates. They're all sugars, right? Sugars can form chains. A glycan is a long chain of, carbohydrates, and it is attached to a protein core. There's a long protein rope with these carbohydrate arborizations coming off of it. Oftentimes, these carbohydrates are heavily charged in this example. All right, so we have chondroitin sulfate. Here's a carbohydrate with a sulfate group. Keratin sulfate, there's another sulfate group, an anionic, an acidic, anionic charged group. Here's hyaluronic acid, or hyaluronate. It's the conjugate base, so here that has been ionized. The point is, carbohydrates. When you take a teaspoon of sugar and you put it in your tea, what happens? Does it sit there and fall to the bottom? It dissolves. Carbohydrates are basically big rings with lots of OH groups sticking off of them, right? Which makes them able to go into solution extremely well. Extremely well. They dissolve. They have an intimate relationship with water, okay? Because they look like water. Here's water, so, right? Forms a hydrogen bond there. This hydrogen gets on some of those lone pair electrons. Carbohydrates get solvated with water and then don't like to be desolvated. Right? Furthermore, these charge groups make the glycosaminoglycan even more attractive to water. These hydrogen bonds form here. Why? Well, these glycosaminoglycans get heavily solvated and then they don't dehydrate very easily. They don't dehydrate very easily. And they make this gel. They make a kind of a gel when they get fully hydrated that doesn't dry out easily. So for example, mucus is a type of fluid that has these glycoproteins in it that holds water and resists dehydration. Saliva, the slime that's on a slug, whatever, any of those kinds of things protect the surface and don't dehydrate very easily. On top of it, these sulfate groups want to stay away from each other. So it also makes it kind of slippery. It keeps it wet, slippery, gooey mess. All right? You don't need to worry about the structure so much, just that it makes it hard to dehydrate. these different structures can be found all over the place. For example, chondroitin sulfate is what makes cartilage sort of stiff, and high hyaluronic acid is found in the bursa of your joints, which makes your joints not grind when they move over one another. It's the vitreous body of the eyeball. It's the clear jelly that light passes through in the inside of your eyeball. All right. Should go into that more. Dense connective tissue. Type of connective tissue property. Here's an example. Tendon is a great example of it. All right? Dense connective tissue. Tightly packed cells, regular in array. They're all going in the same direction. Resists force along a single dimension. Okay. Another type of dense connective tissue is dense irregular connective tissue. So, also very densely packed cells with lots of collagen and very few visible cells. However, it's not resisting force in a single dimension. It is in a single direction. It's resisting force from unpredictable directions all over the place. We find this in the deep layer of the dermis. So the reticular layer of the dermis is got this dense, irregular connective tissue. We have the papillary layer on the top, and then below that, where the hair follicles and the sebaceous glands were embedded, is this dense, irregular connective tissue. Okay. And then finally, elastic tissue. This is the third type of dense connective tissue. It's made of elastin. It's stretchy. So this is part of the example I give here is the elastic ligaments in the back, which enables your vertebra to stretch with respect to one another, but stay in a stable orientation and return to their original position. Moving on to the loose connective tissue. We've talked about areolar tissue already. It's very loosely packed. All right, it's not dense. There's a lot of seemingly empty space filled with this ground substance, with the glycosaminoglycans, with the proteoglycans, etc. So this is underneath all epithelia. It's found in the serous membranes. It's in between muscles. The neurovascular bundles run through this stuff. It's a nice, stretchy, deformable, supportive substrate for a lot of things in the body. Reticular tissue. I already gave you this example of kidney here. So this is very strong, a little bit inflexible, strong in 3 dimensions tissue. It can form the supporting capsule or stroma, the word here stroma, for the various lymphatic organs or a number of other places in the body. So the lymph nodes, the spleen, the thymus, bone marrow. All right. And then adipose tissue. The white fat is very common for fat storage. Can slow heat loss, and then the brown fat that I talked about, that's my girl. They have, it's enriched in mitochondria, which is what makes them, and it's also more vascularized, which makes the fat brown, and it can break down very quickly to release those fat stores. All right, supporting connective tissues, bone and cartilage. Bone becomes calcified, gives weight support, is highly vascular because of the Haversian system. Remember we talked about that yesterday a little bit? We're going to talk about it in lab today. And then cartilage. It's this sort of gel type brown substance that is not ossified. It does not have cartilage. It does not have any calcification. It has these lacuna, these holes in it that allow it to compress and absorb shock. So you find it at the junction of bones, so that when bones come into contact with one another, it doesn't hurt so much. And then it is importantly avascular. Avascular. So this is one of the biggest differences between bone and cartilage. Bone has lots of blood, and you can heal a break to a bone very easily. Cartilaginous tears. Anyone an athlete here or anyone else that's had a cartilage injury before? Yeah. It's a pain, isn't it? You guys are young. You're going to be fine, I guess, I hope. But protect your cartilage. Protect your cartilage. As you get older, it becomes much harder to heal cartilage. Cartilage gets its nutrients by diffusion into that tissue. That's why diet and hydration are important with cartilage. Having I'm going to plug for yoga again here. This is why compression and expansion of cartilage is so important, because when you put gentle traction on cartilage, it helps draw fluid into that cartilage. It's why it's so important, particularly for the soft tissues, to do the right kinds of stretching to warm that tissue up and to bring nutrients into the tissue. That's why yoga, for example, was one way. You have a lot of this kind of passive inversion stuff, so I'm lengthening my spine there, pulling on the cartilage, the fibrocartilage, and bringing nutrients into that cartilage. It's avascular. It doesn't have any other way of getting nutrients to these chondrocytes that are living in those pockets. This is the time of your life to become conscious of it, because when you're in your 50s, the game is pretty set at that point. 40s even. All right. Yeah. Cartilage. So there's this matrix that's made up of the proteoglycans, the collagen, and various brown substance proteins in between. There are the chondrocytes, which are the cartilage cells, the terminally differentiated cartilage cells that live in these lacuna. The lacuna are the little chambers, the holes that make the cartilage compressible. All right, so it looks a little bit like a sponge. It's got all these holes in it. It's more easily compressible. This brown substance is also compressible. It's not been calcified. It doesn't have a hydroxyapatite ceramic in it that bone has, given it its rigidity. On the outside, this is actually not the best picture. You can see just a little bit of it here. This would be a strip of cartilage here. On the outside of it, there's this perichondrium. It's a fibrous outer layer that have the chondroblasts, the cartilage stem cells. In that layer, they will differentiate and then become chondrocytes down the road. All right. Growth of cartilage. I don't want to spend too much time. So there's interstitial growth that happens inside the body of some cartilage. You have a single chondrocyte that goes through cell division, and you have two cells which eventually form some new matrix around them and separate from one another. And then you can also have this appositional growth, where this is the perichondrium, this is the fibrous layer and the cellular layer, and we have the chondroblasts, the cartilage stem cells, begin to divide here and migrate this way into the body of the matrix of the cartilage. All right, I think I've hit the point that it's avascular. This is the most common type of cartilage, hyaline cartilage. It's clear. and you can't see the collagen fibers because they're extremely fine. You'll learn to recognize a hyaline cartilage by the way the chondrocytes look and the lacuna. The lacuna are stacked up a little bit, but they're not like the elastic cartilage where you have tons of stacks of them where it looks really packed in. You find this all over the place. It's the articular cartilage between bones, between the ribs here, this costal cartilage we talked about, trachea, the larynx, the windpipe. The entire fetal skeleton is made of hyaline cartilage because that is what develops into bone. So we talked about intramembranous ossification yesterday. That's what's happening in a fetal skeleton. All right. Elastic cartilage looks quite different than hyaline cartilage to me. Less matrix in here, many more lacuna, much more flexible and elastic, so that your nose, the pinna of your ear, your epiglottis, these are elastic, pieces of elastic cartilage. Finally, fibrocartilage also has a very distinctive look. There's a sort of directionality to fibrocartilage where you have long strings of chondrocytes with much more recognizable collagen fibers laying along that same plane of directionality. It helps to create, in fibrocartilage of the vertebral disc, these annular layers of cartilage that help keep that disc in place when it's put under pressure. Helps it keep from rupturing and allowing the nucleus to move out that way when you put that disc under pressure. So fibrocartilage, very strong compared to the other types of cartilage. Resist compression and absorb shock. All right. Fluid connective tissue is the blood. So the plasma is the watery matrix. So the matrix here is not the solid stuff of the cartilage or the gooey ground substance of areolar tissue. It's just it's a fluid. It's just a total fluid, plasma. And the formed elements are the cells. We have red blood cells, platelets, and then the leukocytes, the white blood cells. And we're going to go through all of those in much more detail elsewhere. All right. Concept of a membrane. God, I should have come up with a picture. 4 types of membranes. Mucous membranes. We find these on, I'll go through them all. Find these, like, for example, on the mouth and openings to the outer world. Serous membranes, we talked about yesterday. Synovial membranes, these are inside the joints, the joint capsules. And then your skin, the cutaneous membrane. The rest of the class is going to be devoted to talking about these 4 types of membrane. So, membrane. That is a membrane. That helps you at all, or no? Mucous membrane, mucosa. So these are lining the passageways that have an opening to the outside world. This includes your digestive system, your entire digestive system, your urinary tract, the reproductive tracts. They need, those surfaces, the mucosal membranes, need to be moist. They need to be moist. They can't dry out, both to reduce friction and to facilitate the absorption and excretion of products across this apical surface. They are supported by a lamina propria. So that underlying connective tissue layer that is supporting, so here's the ET, here's the CT, in mucous membranes, we call that CT the lamina propria, right? And we'll come across that term in a number of places down the road when we talk about the digestive tract, etc. Yeah. The way it stays moist is by releasing these mucosal secretions. Mucosal secretions are, again, these glycosylated proteins. Mucus is a glycoprotein that has all of these sugar chains that resist being dehydrated. They resist being dehydrated. We'll leave it there. Second type of membrane, serous membrane. We talked about these yesterday. These line cavities that are not open to the outside world. They're very thin, but they're strong. And they have both a visceral portion and a parietal portion, and then a cavity in between them, into which the serosanguinous fluid is released. So this visceral portion is also called the serosa. So here would be the heart, and the visceral pericardium is the serosa of the heart. Here's the transudate. The transudate is that serous fluid that is being released, that is allowing there to be very low friction, as for example, as the heart is beating inside of its cavity. There's the three serous membranes we covered in lab yesterday. I should have quizzed you guys on this here. I had an empty slide, so just throwing it up. But we remember those. The pleura is the lungs. The peritoneum is in the intestinal compartment, and the pericardium surrounding the heart. Just leave it at that. All right. The third type of membrane found in the body, tissue membrane found in the body, is the synovium. It lines the joint capsules in any movable joint in your body. So, for example, your elbow or your knee or your hip or finger joints or whatever. We have the articular cartilage. This is some hyaline cartilage here, compact bone. And then in this synovial space here, in this joint capsule, it's full of synovial fluid that is being secreted by this synovial membrane. Here are the epithelial cells. Here is the underlying areolar tissue of the connective tissue, helping to form the membrane. These epithelial cells are releasing synovial fluid that are acting as a fluid cushion to the joint, are lubricating the joint as these two bones are rubbing against each other when the joint is moving, and are also supplying nourishment, nutrients, a nutrient broth for the cartilage, right? So that cartilage is by diffusion grabbing its nourishment from the synovium. All right. Finally, the cutaneous membrane. It's the skin, covers the body. It's very thick, waterproof, and the surface of it, unlike the surface of the other three that we had talked about, is dry, by and large, when you're not sweating. Okay? It is certainly not wet like a mucosal membrane or synovial membrane or serous membrane would be. It's made up of an epithelium, an epithelial layer, with areolar tissue beneath it, and then the dense irregular connective tissue beneath that. So that's the ET over the CT there. We're going to go into some skin in more detail on another slide here, but first, covering the concept of the fascia. The fascia is the connective tissue superstructure of the entire body. So there is superficial fascia that is between the skin and the body wall. It's mostly made up of areolar tissue, adipocytes. This is also known as the hypodermis, or the subcutaneous layer. So it's just below the skin. This is sort of the like, it also acts as a thermal barrier to yourself. Then there are these deep layer of fascia, not invested with fat. Instead, this is dense connective tissue. Is anybody a cook here? Let's cook that works with meat. Cut meat up much, beef. If you ever cut like a top round steak up or something like that, you'll see the layers of two different muscles surrounded by an almost opalescent sheening layer of really pretty tough stuff to cut through. It's not gristle. It's just a thick, strong layer of connective tissue that is this deep fascia, right? And then finally, there's a subserosa fascia that is found between the serous membranes and the deep fascia. So this is areolar tissue. This deep fascia is helping to separate muscles and define ligaments and tendons. All right, that's enough on the fascia. Woo! It's a lot of skin to cover. I don't think I'm going to get through all this today. I went slower than I intended. We'll see where I get. Let's see here. Yeah, we'll do skin. We'll skip the hair probably. Skip the hair and nails. All right, the integument is the largest system of the body. 15% of the body weight is tied up in your skin. That's a lot. That's actually quite a bit, given the dozen or so organ systems that we talked about. Every person's body is about 1.5 to 2 square meters in area, and it's a couple millimeters thick. Not that thick, actually. There is the cutaneous membrane, the surface of the skin, and then all the accessory structures. I think I had said this yesterday. There's thick skin and thin skin. Thick skin is found exclusively on the surfaces of your palms and the plantar surface of your feet. There is one extra layer in the epidermis, and there are no hair follicles. On the other hand, there is thin skin, and this covers the rest of your body. It does have hair coming through it. Just because it's called thin skin, people think people's back is thick skin, but it's not, because hair does grow on your back. So the rest of your body is coated in this thin skin. All right, functions of the skin. It has hot and cold protection. It protects you from thermal damage, from damage from the sun. It acts as a water barrier, mechanical and chemical protection. You do absorb sunlight through it. It acts as a pathogen barrier and pathogen destruction from the white blood cells found there. And then it gives rise to some sensation. It also is important in conveying gestures and emotions. The way that you use your skin, contort your skin, is part of nonverbal communication, as we see there. Anywhere from 80 to 100% of the vitamin D3 that our body uses comes from exposure to the sun. So getting a little bit of sunlight is not a bad thing. You don't want to get burned, but you do want to get some exposure to the sun. We live in a pretty sun-phobic culture, but we do need vitamin D3 in our diets to help maintain proper bone density. All right. The epidermis is avascular. There is no blood in the epidermal layer there. It's stratified squamous epithelium. Any nutrients that get to the stratum germinativum of the papillary layer of the epidermis get there by diffusion from the dermis. So I don't know if there are any James Bond fans there, but I'm going to burst your bubble. In this movie, it was Goldfinger, and they dipped this poor actor in gold, and she supposedly died because her skin could not breathe anymore, and somehow there was this notion that you absorbed oxygen through your skin, and by blocking off all the skin, this person died. That is totally fictitious. That is not true. Don't buy it. All right. A couple slides going over the structure of skin. So here's just some anatomy. Dermis, papillary layer of the dermis, the dermal papilla, the little hills. And the bottom is this stratum basale, the first layer of cells. It's the basal layer, the stratum basale. This is all part of the stratum germinativum. Right? The stratum germinativum are all the cells that look like cells here, that look alive. They are made up of each of these different categories. Stratum spinosum, those germinative cells, the stem cells from the stratum basale, begin to get spiny and pokey, and then as they metabolically wind down, they eventually get to the stratum granulosum, where the cells become granulated. They've made all these waxy components in them that are going to form this dead, waxy surface to the skin. Stratum lucidum is only found in thick skin. And then finally, we have the stratum corneum, which is that outer layer of the skin where the skin is full of dead keratinocytes and has formed this layer that constantly is shedding. We're constantly shedding dead skin cells off of the surface of our skin, usually in small bits that are too small for us to really see. There's a number of elements in here. Here is various stem cells. Here's a melanocyte. This is a dendritic cell, a tactile cell that is a nerve cell that's up there, giving us tactile information. We'll talk about that later in the nerve chapter. Oh, that didn't come out right. Bummer. Well, this is just going through, it's a similar slide going through the time course of the life history of a keratinocyte. So they form from these stem cells in the stratum basale. Down here, this is where we start. And then we go up into the stratum spinosum, which is the long green area here, which is the bulk of the epidermis. These keratinocytes are pushed upwards, and they're just producing lipids the whole time into these granules. At a certain point, they are mature. Their metabolism bottoms out because it's a pretty long way for nutrients to diffuse up to them, right? It's diffusion-limited absorption of nutrition, and their metabolism winds down, they've also been packed full of these oily-filled vesicles that they've been producing throughout their entire time in stratum spinosum. So they're pretty much ready to die. And they release all of their lipids through vesicles. In the end, after about a month, the cells on the top there form this stratum corneum, and it's just this dense, tightly packed layer of dead, desiccated, oily cells. This oily keratin infused layer of cells up there. All right. Beneath the epidermis is the dermis. And this is made up of the papillary layer and the reticular layer. The papillary layer is areolar connective tissue, and the reticular layer is dense, irregular connective tissue. We find the hair follicles embedded in here. We talked about the anatomy of that yesterday. There are various sensory organs here and sweat glands. So all of that is important, but we've already sort of covered that yesterday, so I'm going to keep going. The hypodermis. This is the subcutaneous tissue below the dermis. This is part of the fascia. This is typically where this is always where hypodermic injections are, but this is typically where injections of any sort are. It's into the fat layer that goes beneath this connective tissue layer of dense irregular connective tissue. This is where a lot of fat is stored, and thus it's where there's thermal insulation. Women just genetically, on average, have a slightly thicker subcutaneous fat layer than men. Just means they're better insulated. Hypodermic injections are not the only place you can get injections. You can have intradermal injections as well. They're just not going as deep with the needle. It depends on what your strategy is, what your goal is with the injection. All right, skin color. Skin color is dictated by melanocytes. So here are melanocytes in the stratum germinativum. Here's a cartoon of what they look like. These are cells that have all these pseudopodia that come off of them, these little tentacles that come off. They are able to migrate between the cell junctions of these epithelial cells. They're able to unlock any tight junction that may exist between these keratinocytes. And then they invade the cell and release these vesicles full of the chromophore melanin. You can imagine that already this has a couple elements in its natural function that would be useful if you were an aggressive cancer. Right? They are able to move in between cells, penetrate cells, the junctions of cells that you would normally not want penetrated. Okay? So there's fewer dominoes that need to fall in a melanocyte for a melanocyte to become a dangerous type of cancer. All right. So here is interesting. Different skin colors actually have the same melanocyte density. It doesn't. So someone of an African origin versus someone of a European origin, they have the same number of melanocytes in their body. It just happens to be that the melanocytes in dark-skinned people are making more melanin. They're just turned up. Same number of cells, just turned up. That's it. And they are more diffusely spread out. So their packages of melanin are spread out to more of the surrounding cells, offering these people more protection from the deleterious effects of the sun. Yeah, and the melanin that they produce is also broken down more slowly, so resides longer in the cell. All right. I don't think I was going to ask anything about these, so I'll probably skip this. These are just some pathology, skin pathology. These are definitions of different words. Cyanosis, blue skin; erythema, red skin; pallor is white skin, meaning no blood; albinism is lack of melanin in the skin; jaundice means your bilirubin is high because you're having liver problems; and hematoma is just a bruise, is a mass of clotted blood in the skin. All right, hair, nails, and cutaneous. That was the day before I gave my PhD dissertation. My buzzer broke. But unfortunately, I don't really have time to talk about a lot of hair here. Yeah, or nails. I'm cutting out hair and nails, and even sweat and that. So where am I going with this? All right, let's go here. Sorry, I cut out all the accessory structures. Just went too slow today. Burns. So burns are a real problem. It's the leading cause of accidental death. You may not know that. These are from fires, sunlight, kitchen accidents, acids, bases, electrical shock, etc. So the death typically comes from fluid loss, infection, or other toxic effects of Escher. Escher is burned dead tissue. So here's some Escher. This isn't just scarring. This is dead tissue, the Escher. Fluid loss is a real problem because the skin provides a water impermeable barrier that stops fluid from evaporating off the skin. Here's a first-degree burn. This cool dude has just been sitting out at the beach a little too long, waiting for somebody to notice how cool he is. This usually resolves itself in a few days to a week. And then the second-degree burn becomes blistered and is very painful. This is a burn that hurts. A second-degree burn hurts. This can take anywhere from a couple weeks up to months to heal a second-degree burn. A third-degree burn does not hurt in the same way that a third-degree burn is. Now, remember, any kind of burn like this, there's going to be a gradient. There's going to be third-degree surrounded by second-degree, surrounded by first-degree, right? So this is not to say that someone who has third-degree burn does not hurt, but where the third-degree burn is does not hurt the way it hurts where there is second-degree burn, because you have burned past the nerve endings. You've burned the tissue, okay? So third-degree burns are serious, and those all of the germinative cell layer, skin layer, that had been there has been destroyed, and if it's extensive, the body is not going to be able to have skin tissue migrate its way from the edges back to the center of where that burn was. So often, if a third-degree burn is big enough, which means anything besides a small one, you're going to require some skin grafts. And one of the biggest problems with these people is fluid maintenance, maintaining the proper fluid. So they're on IVs, and there's all kinds of strategies for limiting evaporative loss over the burn, and then infection gets these people. Skin cancer. Well, I'm going to skip over the skin cancer. There's 3 types. Basal cell carcinoma, very common, the least dangerous, the most benign. Squamous cell carcinoma. It comes from the keratinocytes. Also relatively benign if caught early on. Malignant melanoma, it'll kill you. If you get past the stage 1, it's very hard to survive this stuff. it's only 5% of the skin cancers, but 75% of the deaths. So the ABCDE rule, is it asymmetric? Okay, that is definitely asymmetric. Is there border irregularity? There's certainly some border irregularity there. What kind of color is it? Is it not a uniform color? Is there this modeling? How big is it? Is it getting big? And is it evolving? Is it changing with time? So the ABCDE rule, those are the factors to look at when assessing a skin lesion as to whether it may be a melanocyte or not, or just a nevus, which is like a mole. All right, I'm going to skip repair of the integument because I want to tell you, get ready for this one, about this story. This is the pathology right at the end of class, so I made it. Recessive dystrophic epidermolysis bullosa. This is, there are so many things that can go wrong with your skin. It's amazing. But this is one of the more disturbing of them. There's a gene in the collagen 7. There's a mutation in the collagen 7 gene that leads these people to spontaneously form blisters on their skin. So, for example, if you were to come up to a person with EB, there are several types of EB. Some are not as severe as this, but a person with RDEB or junctional EB, you come up to them and you just do this to their skin. That would instantly raise a blister on the skin. And then the problem is the fluid pressure in the blister. You get a blister and it feels tight, right? The fluid pressure in the blister opens up that blister along the margins. Okay, so the margins of the blister are not strong enough to stabilize the blister, and the blisters can spread throughout large portions of their body. The blisters need to be drained to stop them from spreading. And it gets worse. Collagen 7 is found throughout your whole GI tract. These people have to be on soft food diets their entire life. They're prone to corneal abrasions. They are on soft food diets, and because of that, they lack tonus in their rectum, and so have to wear diapers their entire life, which can lead to some pretty messy situations for them. Their bodies are often 80% covered in healing skin at any given time, often covered in bandages requiring daily maintenance. It's a pretty arresting disorder. The reason I'm so moved by this and decided to put such a kind of depressing image up here is for a while I worked at for so many years I worked as a camp counselor at a camp for children with chronic skin disease, and I dealt with many patients that had some just beautiful young people that came to this camp. Not all of them had this skin condition, but some of the more affecting ones did. And so I'm telling this story to tell the other side of it, which is not just the absolute horror of the disease, because it's a horrible thing for these people, but the importance of these people, because the first time I was doing a bandage change on a man named Justin Cross. He was really just a boy at the time. He was maybe 10 or 11, and he goes through this daily process of getting in a bathtub, soaking himself so that the bandages are able to come off of this skin that they have. Because if you did it dry, it would just peel the skin right off of them. You have to soak them first to take the bandages off. And I was doing this to him for the first time. I hadn't done that before. I was pretty intimidated. I have to say I was pretty intimidated. And he's sitting there just wincing, beautiful young boy, had blue eyes. Justin Cross was his name, and he could. I was being really ginger with the way I was handling him, and he stopped me at that, in the middle of what I was doing, and he said to me, "Tom, don't be afraid to do what you have to do. It's going to hurt me. I understand that. It's okay. Don't be afraid. Just do what you have to do. You can do it." And like that moment, the effect it had on me, where I saw that this person whose entire life had an entirely different scale of suffering than I was even aware of, much less having any kind of personal experience of. That person who was so helpless in so many ways was offering me support, was giving me insight, was giving me strength. That was really a profound insight for me. And in a lot of different ways, I like telling the story because it continues to keep it fresh for me, that first of all people have suffering, all people have struggles. No person's struggle is greater than the other, and everybody's unique in their struggles. And those who are so marginalized, who have so little, who have such great struggles, can offer some of the deepest insights, can offer us some of the greatest, the greatest potential for growth. So that is something I hope you can take from that story. There was another woman at this camp here. She died a year ago. Actually, she died a year ago. Before she died, she was the oldest living person with recessive dystrophic epidermolysis bullosa. She was a Mormon, lived in Utah. I met her at the camp. She had just a beautiful singing voice. She had a really beautiful singing voice. And she actually looks, she's in such good shape because people like to rip on the Mormons, but I'll tell you one thing, they are a community with some heart. She was really taken care of exceptionally well by her community. She really had all the support she needed to pursue her dreams and her talents. And very uplifting person. I feel fortunate to have known while I did. So anyways, that's it. That's the end of the story. But the camp, if you're interested, is called Camp Discovery. It's run by the American Academy of Dermatology. And for those of you going to go into medicine, if I could do it again, I would do dermatology. There's a lot to recommend dermatology for. The hours are great. The lifestyle is great. You don't have to be on call so much. The pay is good. And it's just fascinating. It's just a fascinating field. So that's that. Any questions?